Thank you for reminding me. Please, I don't want to use your names, and thank you for reminding me of that. So when we're discussing these cells, you got to remember we're talking about things in 3 dimensions. We're only able to deal with 2 dimensions on this particular on this video. But let me take a break and show you 3-dimensional cells. Recording. So now that we looked at the 3-dimensional structure using the virtual reality game, you see that we have this drawing which is drawn in 2 dimensions. In other words, I only have length and width here. I can't show you depth. And you should know that the cell is a sphere, right? It's all the way around. And I can't even draw that. I can draw it as a circle, right? But it's 3 dimensions. So don't think of this, even though we're talking in 2 dimensions, because that's what I can draw, you have to remember that there are 3 dimensions to this. So here's this membrane. The only things that can get in and out are very small nonpolar molecules. So something like oxygen, it's tiny, and it can easily fit through. Oxygen can go through the membrane with no problem. CO2, why? Because it's nonpolar. And it's small. So it goes into water, it diffuses into the blood, and you can easily go into and out of the cell, no problem. CO2, remember this is inside, and this is outside. So one of the primary uses of the membrane is to separate inside from outside. I mean, I think that's obvious, but just to make sure you understand, the membrane separates inside from outside. That's what the cell membrane does. But like we just talked about, it can't just let things in and out. CO2 is also small and nonpolar. They're very tiny, these molecules. CO2 goes out into the blood. That's why we exhale it, right? And when we inhale, we're taking the oxygen into ourselves, and that means it's going into the cells, right? And it all diffuses. In other words, it doesn't need a protein. These small nonpolar, so let's write that down and keep a list. Small, nonpolar, they don't need any help getting across the membrane. Not at all. They go back and forth with no problem. The things like sodium that are charged, things like potassium, those things are big enough that they need help getting across that membrane. Does that make sense? Yes? Okay. Yeah. Any questions about that? All right, so let's... All right, question?
Small, nonpolar, and not charged, they can go in with no problem and no help.
That's right. So that's it. That's the key. Small, nonpolar, in and out. You'll find out how. Someone asked this morning a really great question. Well, what determines whether it's going in or out? Well, we're going to learn that. It's called diffusion and osmosis. We're going to look at that. We're going to take some eggs. We're going to dissolve the shell off the egg, and then we're going to use osmosis and diffusion to swell up the egg or shrink it down. So we'll get water to go in or out. It's a cool experiment. Not hard-boiled eggs, so we have to use our gloves, right? So here we got, there's a cell membrane. So as we just said, the membrane is used to separate inside from outside. The hydrophilic tails are pointing towards the water. Where's the water? Inside and outside. The only place we don't find water is in the middle, right? Very little water. Sometimes you get a water molecule in there because water's kind of small. Even though it's polar, you might get some water going through. But it doesn't like water in here. Nothing polar. Certainly nothing big. If it's a big thing, it's not going to sit in there. Not going to be able to pass. So if you see something like sodium and Na+, sodium ion, or potassium ion, or calcium ion, or a chloride ion, if those things are not, they try to get past here, they can interact with water, no problem, but they can't get past this membrane. So they need a what? They need, no, They don't need no, carbon and, not carbon and oxygen. Yeah, maybe you missed the point there, but when I said to you that this layer is nonpolar, when I say to you that oxygen gas and carbon dioxide gas, those are molecules. They pass by easily. All right. These are sodium chloride and potassium. They're charged. They won't be able to pass through. But you need them inside the cell. You need to get them inside the cell. So you have this barrier. How do we get them past the barrier? The pores. We need to have doors, right? And if we think of the cell membrane as a wall, what does every wall need in order to get things inside and out? You need a door. So we need doors. We need windows. If without openings, there's no good. It doesn't help. And of course, it's not that simple. It sounds pretty simple right now, but it won't be that simple in a minute. So we need some kind of door. What are those doors in a cell membrane? What makes those up? What's the name? What class of what's the class of biomolecule that makes these things up? You just said it earlier. Proteins, that's right. Proteins. And yes, they're channels. Yes, I agree that this example is a channel. There's other kinds of things that let things in. There's different kinds of doors, right? You have doors that swing both ways, right? You have revolving doors, don't you? You have automatic doors that slide open and slide closed, don't we? We have many different kinds of doors. And there's many different kinds of proteins that go in the membrane. That's why all those proteins were different. The ones that you built in your model, they were all different. This one was the channel protein. And if you paid attention to it, what you saw was a dot in the center, right, with an arrow pointing in this direction. What do you think the arrow meant? What do you think they meant by that arrow? Say that again? That's the way it goes. It's trapped. That molecule. In other words, the point is that these calcium, sodium, chloride, potassium, whatever, they can get through this protein. Just like we just saw the little spaceship go through the membrane. This protein is what allows this membrane, these things that can't pass through otherwise because they're charged, get past the membrane because they create this opening in the membrane. Do you see that? We're going to try to do that with bubbles. We're going to create these little strings. We're going to take little strings, we're going to make little openings, and we're going to create an opening in the membrane. It's going to be kind of cool. I think it's one of the coolest experiments we do, but it's more of a lab because it's not really an experiment. So let me ask you this. If this is sitting in the membrane, remember it's three-dimensional, so we just cut this open, right? So when you're looking at that, this is cut open. So think of an orange, and you slice an orange, and you look inside, right? That's what you're doing with this membrane that I'm drawing up here. So this is three-dimensional, so it's actually more like a tube or a pipe. Can you think of this as a pipe? Do you see that? Can this be a pipe in your mind? Can this go all the way around? In other words, what you should be seeing is something like, it looks like this. Right? It does, a little bit like a macaroni. So it's more of something like this that's sitting there, right? Where things can go into this tube and then come out the other end. Do you see that? Do you see that there's an outside? Can you see that there's an outside? That's this side here. And then there's an inside. That's the inside of the tube. Do you see that? Okay. So if we say green is, let's say green in this key. Let's create a key. Green equals hydrophilic or polar, right? In other words, water loving, right? Hydrophilic / polar. then it makes sense that all this here would be hydrophilic because it's sitting in water. All this is water out here, right? And all of it's water out here, in here, right? We agree? We just saw it in a three-dimensional cell. And if it's letting things through, then this must be hydrophilic as well, inside, right? So the inside must be green as well. But what must the outside be? It must be nonpolar. So in this color scheme, what must the outside be? In this color scheme that we see up here. Yellow, very good. So this part here must be yellow because that's the part that's sitting in the membrane. Do you agree on that? Does everybody disagree? Someone not understand? Please ask a question now. It's okay you don't understand, but ask a question now. So that part has to be yellow. Does that make sense? Because this part here has to be yellow because it's touching the hydrophobic portion. So as we're looking at the outside has to be hydrophobic, but the inside has to be hydrophilic. Does that make sense? So that's why I asked you to draw it like this. If you saw yellow, when you did this on the project, I asked you to make sure this was the one color, the hydrophobic color, and I asked you to make the other part the hydrophilic color. Did you see that? That's why I wanted you to do that, so that you can start to understand what we mean by these membrane proteins. Remember, this is a protein. So this is one giant chain or a couple of chains of amino acids, right? And there's 20 different amino acids. And these 20 different amino acids, some of them are polar and some are non-polar, right? Yeah.
So this is like kind of like a 2D version of
That's right. This is a two-dimensional version of what you guys did and built already. Does that make sense? So hopefully you guys are able to see that this is you're able to see that this membrane protein allows things in and out of the cell, and that you have to have a nonpolar end and a polar end. Are we all good with that?
Yeah.
Do I expect you to know the names of all the different proteins? No. But you do have to know what they do, right? So one function of proteins on a cell membrane is to let things in and out. Do we all agree? And by the way, I guess since we did green, we should do yellow, huh? Yellow equals nonpolar. Is everybody okay with that so far, so I can go on? So the little things, little nonpolar objects like CO2 and oxygen, they're nonpolar, they're small. They can go in and out of the membrane without problems. But if they're big nonpolar objects, or they're charged, or even medium-sized polar objects, they can't get through without help. They need a protein. You need an opening, just like the little spaceship couldn't get through the membrane without a membrane protein. These are called integral membrane proteins. Integral meaning they're part of the cell. The integral meaning they're part of. Let me pause this. Okay, so there we have it. There we have our integral membrane proteins, and we understand that that's the one of the functions that allow things in. Now let's take a look at this little thing called the blood vessel. Those of you that went to anatomy camp know what that blood vessel is. Actually is made of what? What is everything in your body made of? Well, yes, there's a lot of water, 70% water. That's good. But what is every part of your body, all the parts of your body, what are they made of? Cells. Somebody said it, cells. Yeah, that's right. And inside the blood, now what goes in here is what? Blood. So blood is in these pipes, right? The pipes themselves are made of cells, right? And blood is what's in it. What's the blood made of? Let's make a list. What's blood made of? Blood is a tissue. It's a tissue. What's it made of? Come on, give me, some ideas. Huh? I want to say, first I want something that you all know what's in there. Hopefully no bacteria. If you have bacteria in your blood, you're in trouble. You're dead or dying. Blood cells. Good. Blood cells. What kind of blood cells? White blood cells. I was looking at the color black on my screen this morning. I said black white cells. No, not black white cells. That doesn't make sense. White cells, and what's the other one? Red blood cells. So white and red blood cells, definitely. The red carry the oxygen, the white are your immune cells. There's at least two kinds of cells in there. What else do we have in there, in that blood? There's a lot of stuff in there. We're only going to list a few things. Huh? Proteins. There are definitely proteins in there. Amino acids, right? They're carried usually in vacuoles, food vacuoles. We know that. Your body doesn't just have random stuff floating. Yeah. What else is in the... Come on, you guys have covered diabetes. You know what else is in that blood. Sugar, glucose. Glucose is in that blood. You have doctors measuring it, yeah. Iron, excellent. Iron's another big one. There's other stuff, right? There's potassium, there's sodium, there's all kinds of ions, minerals, right? So there's all kinds of stuff in there. There's glucose, there's proteins. There's something everybody forgets. There's also oxygen gas, isn't there? Because that's what, you breathe. That gas doesn't go right to your toe, right? It has to first get sucked up into the blood. And then once it gets in the blood, then the blood takes it to your toes, right? So oxygen has to be delivered to your toes. That's the job of what blood cell? A white blood cell. Oxygen red blood cell. Yes, I'm glad you thought. Red blood cells carry the oxygen. The white are the ones that deal with the immune system. But then you have to get the carbon dioxide out, right? Right, getting that carbon dioxide out. My little toe just made some carbon dioxide. It just made it. Right now. And now. And now. And now. And now. It's constantly making CO2. How does it get the CO2 out of its toe area and out into the air to your lungs? The blood. The blood first, then you breathe it out. That's right. So blood is the next step. So CO2 is in the blood. It's dissolved in the blood. Actually, it's complicated. There's other steps, but we'll just say it's in the blood. It's not CO2 by itself, because if it were CO2 by itself, then our bodies would become acidic really quickly, and we can't handle that. So we do it a different way. But CO2 is in the blood, and that way the little blood cell in your little toe on your left foot can get that CO2 out of there and out to your lungs, right, and out into the air. Does everybody understand that? So that's kind of the circulatory system link. That's what blood is. There's for sure. Water is what? What is water? These are all the solutes, right? These are all the things dissolved in the water. But what is the water? The solvent. The solvent in blood is water, H2O. Water's everywhere. 70% of you is water. So all the other stuff is dissolved in the water or suspended in the water. Blood cells aren't dissolved, but they're suspended in the water, right? Everybody okay? That's why water, the movement of water is so important for life. That's why we're going to spend a couple days talking about this thing called osmosis, which is the movement of water. And diffusion is the movement of everything else. All right, but before we get there, let's finish talking about this. You got to get this stuff from your blood into your cells. Now let's talk about something else. What about communication? So what is this? Why did we just talk about this? Listen, this is one of the big things we got to do. We're going to have to spend a lot of days discussing this. This is transport. This is one of the major functions of your cell membrane, getting things in and out of the cell. Selectively. We do it selectively. Guys, we have to control what goes in and out of the cell. The membrane does that. It does it selectively by controlling what membrane proteins are in the membrane and which aren't. So if you want to let glucose in, you better have a protein that lets glucose in, right? Almost every type of molecule has its own door. Now, let's imagine we have most people are tall, skinny people, and the doors to their rooms are tall and skinny. Would I be able to get through that door? No, I would not. I'm short and fat. So that won't work. So I need more of a hobbit door, don't I? I need a round, short door. I'm okay with that. But that's what has to happen. So every molecule has its own door, depending on its shape and size. Does that make sense? Not every single molecule, but every type of molecule. Yes?
Wait, so the
holy water, holy doors and cell.
Yes, in a way. It's the shape. It's really about shape and size. It's almost like a lock and key more than it is a door and size, but that's it. This is an analogy, right? So yeah, you're right. It's the shape and size of the molecule, the polarity of it, and all the other qualities of that molecule define the kind of door it needs. Does that make sense? The kind of protein it needs. But yes, you're absolutely right. So transport. But there's another function of a cell membrane, and that's communication. So we're going to spend some time talking about communication. The boys in the room may not like it, but it is part of biology, so you're going to deal with it. So let's talk about ovaries. There's 2 ovaries in a female. Yeah.
Before we get to this, did you say that there was oxygen in your blood, right?
Yes.
And I thought your blood turned red when it did oxygen.
Yeah, everybody thinks that, yeah.
So what color is yours?
Hold on, can I just talk? I will gladly explain it. Unfortunately, a lot of middle school teachers are still explaining it this way. It's not true. And, to be fair, a lot of them are confused too. If you ever give blood, and I hope you do because people need blood. So if you give blood at the Red Cross, they'll stick a vein, or if you've given blood, you've gone with your parents to the hospital and they've given blood, they'll stick a needle in a vein, and they will pull out deoxygenated blood. If you look at that blood, it is not blue. It's dark red or black. It's almost black. So I know it's weird, but color, we talked about color a little bit. Color is complicated. So when blood is deoxygenated, it is never blue. All right, only insect blood is blue because insects carry oxygen using something called cyanide, completely different from humans. Humans use iron. And what color does iron turn when it's mixed with oxygen? You know this because you call it rust. It's red. So, a brownish red. And so if you cut your skin and you expose the blood to the oxygen, it does turn red. But it was never blue. Okay? It's just, the reason it looks blue through your skin is the skin changes the color. Yeah. Great question. If you're anemic and your iron levels are low, your blood is still red because you have to have some iron or you'd be dead. So anemic, there's all kinds of reasons for anemia. Anemia is a general category, which means you're having trouble transporting oxygen. It could be you don't have enough red blood cells. It could mean you don't have enough iron. It could mean you have some kind of cancer. I mean, there's all kinds of reasons why people are anemic, right? One of them, for ladies, is sometimes they bleed too much during their periods, and that could actually cause them to become anemic. So yes.
So my auntie, she had an accident. She got shot, but she became anemic. So what did that do?
So there are many reasons why, and I hesitate to tell you anything about it because it all depends on what happened to your aunt when she got shot. So when see, it could be a lot of different factors there. Okay, so I'll tell you, it depends. We need to study that case a little deeper, and I certainly wouldn't like to discuss it on record. Yeah.
Because of what you said, in our discussion, like at work group, that you weren't gonna start anything and not be able to finish it, we have cream.
Uh-huh.
So can we have enough time for this?
I think so.
Okay.
So when we have 2 ovaries, a woman every month releases an egg. A woman every month releases an egg, female, after puberty. So on average with women, girls in our country, I think a lot of girls have their first period at 12. That's the first time they released an egg. Now, an egg is released from an ovary. It literally shoots out of the ovary. And yes, it can release more than one. That's one way you get fraternal twins. There's other things that can happen. We're not going to discuss it. As soon as that cell, that's a cell. When I say an egg, I mean a cell. We call that cell an egg. All right? It's not an egg like the ones we eat. Those are a little different. This is a cell. It's half of you, right? Half of you was an egg. The other half was a sperm. they come in when sex occurs, and you have a fertile female, you implant. As soon as that egg cell is released, it signals the female body, get ready for my implantation. So this thing here is called a uterus. Females have a uterus, and the uterus is where what will become the womb. That's where all of us spent our first 9 months, in a uterus built by our mothers. As soon as the egg is released, it tells the body, build up a vascular tissue. That means blood vessels, arteries and veins. Build them up, so it builds them up. See how thick they are? What are those made of? Cells, arteries, veins, connective tissue. What's inside arteries and veins? Blood, that's right. If that egg, after a week, it travels along here for about a week. After a week, if nothing comes of it, nobody's there to meet it, no sperm are there to meet it, it dies. It stops signaling. So the one. That's you're going to be a homework question. That's why I was going to leave it for homework. Listen, let me finish, because I want to keep to our time. I'm trying to keep you honest now. You guys need to stop interrupting me. Listen, when that egg dies, the signal stops. And what happens to all these blood vessels with blood? They get put out. Shh. They get put out. What do we call that? What do we call that?
Period.
Your period. So your period is actually about a week after you release an egg. And the reason is that the signal is no longer coming from the egg to your tissue here in the uterus. Your homework tonight is, how does that cell signal the woman's body to prepare the uterus for implantation? Leave the binders. I hope you better write the homework down. It's due tomorrow. Write it down. The homework again. You know what? I'll send it to you in Jupiter grades. You do, but you have to have it done by Wednesday. Put your binders down here on this lab bench.
How are you gonna take it?
Let me record this. So this is the membrane. I'm just drawing a white line. And on one side of the membrane, you have particles. It could be anything, right? They're dissolved. And we went through that. We talked about what dissolving means, right? And so they have all these particles on this side of the membrane. On the other side of the membrane, you could have some other particles. And on this side, you have some particles. You see they're green and white? Okay, so on which side of the membrane is, let's say, the white is equal to sodium, positive ion. So the white dots are sodium ion. The green dots are chloride ions, so sodium chloride, right? Table salt. They're separated in the solution. On one side of this membrane, you have one ion. On the other side, you have the other. On which side is sodium ion more concentrated? Let's call this. I'm sorry, that's a good point. Let's call this outside, and let's call this inside. Yeah. We're talking about how things move across the membrane. This process is called diffusion. This process is called diffusion. I'm going to say it again because people say I didn't say transport. I'm saying it right now, it's diffusion. This is transport. This is how things get moved. How do things move? Are there little machines that move things from one side to the other? There are machines, biomolecular machines in your cells that move vesicles. That's true. But we're talking about how things move across the membrane. They move by something called diffusion. Could someone close that door? Can I go to the bathroom? Yes. Yes.
So, well,
Any movement is transport. Right now, we're talking about the kind of transporting across a membrane, movement across a membrane, that we call what? Diffusion. Diffusion, I'm about to explain everything, so let me go ahead.
What's the difference between a transport and a channel protein?
A channel protein helps transport particles across the membrane. It's part of the mechanism. It's part of, transport. You get transported here, don't you? How do you get transported here? A car. So what helps you get transported? The car. So transporting particles across a membrane, what helps transport the particles? The channel protein, the proteins. Do you understand that? So the proteins help move things across the membrane. The membrane tends to stop things, except for what? What is not stopped by the membrane? No, charged particles are. You get that straight.
Not the ones that aren't charged.
The ones that aren't charged, which are what? Oxygen is an example. Oxygen gas is an example, not the only one. Oxygen gas and carbon dioxide gas are the two big important ones. But that's because what quality of these two of gases is it that allows them to cross? They're small and nonpolar. All right. So let's again talk about this. So where is the white highest concentration is where?
Outside.
So white has a higher concentration outside, higher. And I'm going to put brackets. And brackets mean concentration. You're going to have to know that in chemistry. You'll have to, and I'm telling you, I'm going to use it here a lot. It's much easier than writing out the word concentration. So brackets, if you see a number like this, what is it? It's the concentration of whatever. In this case, 4 milligrams per liter, right? All right, so if this side is higher in concentration, what's on the inside? Lower. Does that make sense? So this is going to be, I'm going to tell you right now, I've done this so many years. No, the white. The white is higher on the outside, but it's lower inside. This is the biggest problem kids have on this test that you're going to have next week. I'm telling you the mistakes so that you don't make it, but I can't tell you how many years people still don't resolve this in their minds before the test for some reason. If it's high on one side, it has to be what?
Low on the other.
It has to be.
So higher is greater than the inside than the outside.
In this example, it could be either way. Somebody, hey, Shaw, get up and close that window, will you? I don't know why he has to have that door open. I have to listen to him every day. I'm going to have to talk to him about that. I can't deal with it. It's just too noisy. He has that fan on. So now the other thing is, let me ask you this. With when we got the other side of the membrane, we got the green. The green is what?
Higher inside.
It's higher inside. Do you agree?
Yeah.
So that means just simply by what we just talked about, what does that mean for outside?
Lower.
Does that make sense?
Yes.
I mean, literally, that's all there is to it. The other thing that freaks people out is when we start using words like glucose, sodium, but if you understand dots, if you can understand the dots and how the dots are moving, then you can understand, it doesn't matter what they tell you on the state exam. If they put, glucose or sucrose or galactose, or they give you some fancy word, it doesn't matter. But the only thing that matters is that you understand it's higher over here, it's lower over here. If you can understand that, now you've got footing to be able to answer questions on transporting things across the membrane. Remember, this is only one way. There's many, many ways we're going to cover before the exam. Yes?
Like in the membrane, are there going to be like different sized gaps? How you said different.
Really good question. Let's write it down and let's table that. Because the short answer is no. The long answer is yes, there are different sized holes, gaps, openings in the membrane, and that does impact. But we're not gonna talk about it, because it's not really something it's gonna help you understand transport. All right, so do we all understand this so far? Because if you understand this, then we can talk a little more, and you'll understand the rest. Now, you understand it now because I drew it and I explained it. What's going to happen when you get home is you're not going to remember what the heck you just said. What I just said, rather. So you really need to study this material. All right. So here you have a membrane. There's a lot of stuff over here. And how can I represent how much that outside has higher concentration than inside? Well, I can put more dots. That's right. Good. I can put more dots. I've seen end of course exam questions with just dots. It's certainly, you should know. And remember, concentration is how many dots per milliliter. You have to assume that both sides have the same amount of, right? So, of solvent, right? So we're looking at solute, right? So when we're looking at the dots, what are we looking at? The solute. And it could be sodium, or it could be glucose, or it could be anything. So solute, right? So outside and inside. All right. Now here's the rule. This is the rule. If you remember it, it'll help you get the right answer. But there's many steps you have to memorize in order to get the right answer every time. Here's the rule. It's really super complicated, and everybody gets confused. So here it is. You ready? Everything right? Everything that moves by diffusion goes, here it is, this is the hard part, from high to low. Wow, I know that's real hard, isn't it? That's it. I hope it's easy. It's easy now, but what happens on a test? Who can tell me you forget, and that's really kind of key. You freak out, you blank out, you get them confused. I mean, literally, I can't tell you how many times I give kids questions and I'm like, this is an easy one. I was confused. I don't know what's going on. Everything that moves by diffusion moves from high to low. This is a universal fact, all right? Everything's going to, so in what direction, and you know, obviously the test questions aren't going to be that easy, right? You've had enough of my questions to know. So I'm going to write the movement in red. So what direction are these white particles going to move? Assuming they can move. Assuming they can move across the membrane, because they can't all, not, right? The membrane's selective, right? So they might not let them through. They don't have the right proteins, and so it won't let these particular particles through. So assuming they can move, what direction is that solute going to move? To the left. That's it. Done. That's the first step to answering the tricky questions you're going to get. If you can remember high to low, and you can remember to ask the question. Here's the question you always have to ask every time. You have to ask this question. Does the material, let's say the solute. Can I say solute?
Yeah.
Solute cross the membrane. That's the only thing you have. That's the first question you have to ask. Does it cross? If it does cross, then the solute's going to move from what? From high to low. And that's it. That's all there is to it.
It only crosses though if it's selected.
It only crosses if it depends. What's going to if it's polar, if it's non-polar and small, it'll cross almost always.
If it's polar, non-polar, it goes.
If it's non-polar and small, I think that's like the 5th time I've said it, and I'm recording that I'm saying it second period for the 5th time. Small and non-polar moves across the membrane easily without any help.
Positive, when you say charge.
But positively charged or big things need help. They need proteins to help them move across that membrane. They can't fit. Remember, it's polar, non-polar, so things that are polar won't get past the lipid layer. Remember, the lipid layer is non-polar, won't dissolve it. Acts as a barrier. Like dissolves like. That's right. Yes, you may. Sign up. All right, so things are moving from high to low. Let's go on and answer and move forward. Okay? All right. So now let's take. Wait, it's 10:50. We're out of here in 7 minutes. I don't want to run out of time. So somebody give me a. Let's see. What should we do? Actually, I'm going to give you a problem. I'm going to give you a problem. You're going to work on it, and then you're going to turn it in, and you're going to prove to me you understand. You're going to do a problem right now, and then you're going to turn it in, and I'm going to staple. I'm going to grade it. Let's call it a quiz. So you have this cell floating in the blood or in a beaker, and let me redraw it so it's a little easier to see. So you have this cell, you have this baggie, this cell membrane or a membrane in a beaker. And here it is. On the outside, you have this situation. And on the inside, you have this. I'm going to make it easy for you. I'm going to say outside is 20% solute. So what 20% means is 20 solute molecules for every 100 solvent, right? So we just easily write 20%. It's much easier. On the inside, I'll write 10%. All right. Draw that out, and then draw an arrow, and please cover your answers. Draw an arrow in the direction that the salt, and let me answer this. Let me say this. What question am I going to ask? What's the question I'm asking?
Does the solute cross the membrane?
And the answer in this case is yes, it does cross the membrane. How would you know? Somebody would have to tell you. But you know what they could tell you? Here's a funny thing. They don't have to tell you that it crosses the membrane. Here's what they can do. Here's what they do on the state exam. This is what I'll do on my test. I'm telling you right now. What I'll do is I'll say, hey, the solute, is oxygen gas. So I don't have to tell you yes, do I? Because you should know that if it's oxygen gas, then it does cross. But now what if I change? I can leave everything the same. That's question number one. There's question number one. I'm going to ask you a bunch of questions right now. Here's number one. That was question number one. Number two, what if the solute? So draw the arrow, or right here you can put your answer. You can draw the arrow, but you should also put the answer. In which direction, in or out? This is in, right? This is out. Does everybody understand that? So what direction does the solute move if it's sodium ion? Assuming there's no proteins. Assuming there's no proteins in the membrane. Is the sodium ion, what's going to happen there? And the third one, what if the solute is
We gotta tell.
very, very large molecule, a large molecule? What's going to happen there? So that's three questions.
The spin question is based off this, like, salute across the membrane, right?
That's, well, it's first, for all three questions, the first thing you do is start with this. For this question, ask yourself that question, and then you have to know what? After you know that, what else do you have to know to answer this question? Who knows? Anybody here know? But how do you know the direction?
By the concentration.
What rule? High to low. High to low.
Right, I don't want to.
They're on the board. So the first question one is solute. What if the solute's oxygen? What if the solute's sodium? What's the solute is large? What if the solute's large? 20% outside, 10% inside. Does the first question you ask is, does it cross the membrane? And then so the second part is, if it does cross the membrane, in what direction is it going to move? Is it going to move in or out? Is the solute going to go from the inside here out, or is it going to go from outside in? Which direction is the solute going to move? Are we clear?
Yes.
All right. So now these notebooks, I'm going to keep recording because I just don't know what else to do. I don't want to get another discussion about what I say and what I don't say, so I'm going to keep recording. So I don't know what to do about these notebooks. I fear if I put them in, it's going to...
Kill us.
No, it won't kill you. It'll hurt your grade. So what I'll do is, if you have a notebook that you would like to turn in and it's a good notebook, I'll grade it. If you don't have a notebook that you want to turn in, then I won't grade it. Here's what I'm going to say to you, though. I'm saying it, I'm recording it, I will post this on YouTube. So we will all know what the truth is. And I'm going to put in my notes, in my personal notes, that on October 12th is the day I said this to you. So I don't get into the kind of discussion we got into this morning. I'm going to tell you this. The next notebook collection is going to happen in 3 weeks. 3 weeks. It's going to be a substantial part of your grade. I want to see blue sheets filled out. I want to see notes for every day. I want to see 1 page for every day. I want to see evidence of all 6 R's. Am I clear? Evidence. That means I have to see it. Not you telling me you did it. I need to see it on that paper. I gave you examples. I sent you 2 people's notes. I sent you Miss Moore's notes, and I sent you Angelina's notes. There's absolutely no reason why you don't understand. You should be confused on what the expectation is. But I'm going to give you this one last break. And then after that, you and your parents tell me, hey, my kid didn't know. I'm going to tell them very clearly, here's this YouTube video. Look on this number in the YouTube video, and I'm telling them clearly what the expectation is. Yeah. And either one, it's time to go, though.
For the 3 below slides, are you checking from like that 3 weeks to like the past?
When I do grade notebooks, I'm grading the whole year's notebook. Every time I'm looking to see if you've improved it. I have sent you all the notes, and I'm still going to go and photocopy all the notes from somebody else, and scan them and send it to you. Periodically, I will do that. So if you missed the day's notes, you have a copy of the notes that I believe were a good set of notes taken for you. If you have the membrane model, give it here.
You got Sharpie? So my name is.
You want these questions?
No, I had 8 Sharpies. The kids took all of
Did I do it the
them.
right way?
Look at this. You know how full my pen
Did I
box?
do it the right way?
And I said I wasn't going to lend them out, right? Didn't I say that?
Okay,
Now
can I
look
use
at it.
that to write my name on
No, look at how many are left.
Miss
I had a
Rita,
whole
I'm
pocket
standing
full.
right in front of you.
Yeah, they were too.
I'm not gonna take you.
No. All right, we're done recording. So let me start again. I'm recording, so again, I'm not going to use names. So let me start again with like the steps, because one of the problems people had is I told you that there's a question you have to ask, right? When I did this formative assessment I gave you yesterday, right? I asked you to answer a question, and one of the questions that you guys confused, one of the things you confused is that there's a couple questions you have to ask. A couple questions you have to ask. You have to ask yourself these questions every time. so that you can answer the question you're being asked. So there's going to be a question, the state will ask you these questions, I will ask you these questions, and then you're going to have to ask yourself 2 questions in order to answer them. Are we clear on that? All right. So I'm going to walk you through what it is. Now I'm going to walk you through the problem set again. All right. So number 1, you have a membrane. Now they're going to tell you something about the membrane. Something. It might be something they'll tell you either by drawing it, or by naming it, or they'll describe it. One of the 3. So in this case, for today, up until now, it's going to get more complicated in just a little bit. But for up to now, what we've been talking about is a membrane. And we know that membranes let things in and out selectively based on what? Based on charge, good, and size, excellent. Excellent. Thank you, Jesus. All right, so size and charge, yes. Right, polar and nonpolar, that's good. Yeah, excellent. But that, polar and nonpolar, it's not charged, but that's good. Yeah. So polarity, right? Size and charge. Are we all good with that? So kind of the three things you're looking for, because you keep an eye out for. What has to be in the membrane if it's going to let something in that's nonpolar, that's not nonpolar, and not small, right? So if it's large, if it's charged large or polar, right? What is it going to need? It needs a protein. Needs a protein. Love it. So you guys did learn something yesterday, just didn't get that last little bit, huh? That's what I'm hearing. Is that right? Okay. Oh, makes me feel so much better. I can't tell you. I'm sorry, I just had a wave of goodness come down. I don't know where it came from, but all right. So membrane. So it needs a protein if it's large, charged, or polar. All right. So say we have this membrane. Let's say there's no proteins in it. I'm going to tell you right now. For now, we'll talk about what happens when there's proteins in it, but no proteins. All right. And we have this beaker inside this membrane. And next week, what we're going to do is we're going to create bubbles, and we're going to put holes in those bubbles.
They'll pop.
And no, they won't pop. You'll be able to see the membrane. The bubble is a bubble. When you blow a bubble, that's a type of membrane. It's a lipid. It's a partly charged, partly nonpolar, polar. It creates a sheet. And you see those big bubbles? Those are all giant membranes. Now those membranes they're only a single layer, they're not double layer like a cell membrane, and they don't have any proteins in them. But what we can do is we're going to put a little string in it with a hole, and you're going to be able to see a hole. You're going to put a hole in the membrane, keep it a membrane. And I'm not talking about on water, it's going to be a membrane. You're going to put a hole in it, and you're going to be able to move things from outside to inside. So that's kind of cool. Yeah, the actual bubbles, yeah. I'll show you. It won't pop. You'll have a hole in the bubble, you'll not pop. And I'm telling you, we're going to do it, so we're good. It's actually amazing, really, is what I find. And I've done it for years, so. Anyways, you have these membranes. In this case, the membrane's in liquid, right? On one side, it's liquid on the other. So now somebody asked a really great question. It was a really great question. It is confusing, so I'm going to take a minute and explain it. If I tell you it's 50 milligrams per milliliter out here, and I tell you it's 20 milligrams per milliliter out here, inside rather. So that's inside. 50 milligrams of what? Who cares? Well, it does matter. It does matter eventually, but right now it doesn't. We're not talking about that, right? Okay, I just want you to understand concentration, right? So we have, and you should have some idea about it out here. So there's these dots, right? 50 milligrams out here, and then 20 milligrams per milliliter in here. Now, the question that someone asked a minute ago, which is a great question, it's a fantastic question, I loved it. The question was, wait a minute, are they not there's more out here in this space, right? Outside, there's more outside, more space outside, isn't there? Than inside? Then I don't understand, how can we compare the concentrations that they have if there's more space outside than inside? We standardize it. We create a ratio. That's what this means. There could be 100 milliliters out here, and there could be 10 milliliters in here. But for every 1 milliliter out here, outside, for every 1 milliliter, there's 50 milligrams. So how many milliliters are there out? How many milligrams are there on the outside? So for every milliliter, there's 50. How many milligrams are there outside? If there's 100 milliliters. 5,000 milligrams is correct. Because for every 1 milliliter, you have 50. Outside, you have 100 milliliters, you have 5,000 milligrams. You see what I'm saying? So this is tricky. This is hard for even some older people to figure out. All right. So this is not something that you should feel bad about if you don't get it right away. But you do have to understand that the amount of space, when you're talking about concentration, what it's telling you is for a certain amount of space, you have this much stuff. So if I say 50 milligrams per milliliter, every milliliter is there's 50 milligrams. For inside, there's for every milliliter, there's 20 milligrams. The amount of milligrams, how many milligrams, and how much is irrelevant because we've standardized it. We've reduced the number to the simplest ratio. We all have that? Understand that? That was a great question. You see how great that question was? All right. Now, let's go ahead and do the membrane. Right now we're working without protein, so it's just a phospholipid layer. On the outside, we're going to have, let's make a beaker. And they love beakers. They love to do beakers. And there's your liquid. I say they, I mean the state. They love beakers. They love to put cells out there. So you have this thing, and it's outside you have these things. We'll make little triangles. So there's a lot of ways they do it. They do it just with counting, right? And they also will do it with numbers. So we'll in this class we'll do it in both ways. And they'll say something like, assuming the volumes are equal, right? Assuming the volumes are equal, so the space is equal. Which one of these two is more, of course this is outside, everybody understands that. Which one is more, which one has, for the triangle, which side is more concentrated? Inside. Is higher. Very good. So now for the circles, which one is higher? That's right, good. I'm glad you reminded me. Thank you. So higher concentration, right? We're good? That's simple, right? And I've seen questions like this on many standardized exams where they just keep it simple. But then what they could do is that's one way of doing it. Then the other way they could do it, and they don't have to even give you, they'll do this, well, they'll do other ways as well, but let's go ahead and draw another way. They'll give you another beaker, and they'll give you this, and they'll just give you numbers. They'll tell you this inside is 20, and okay, so inside is, inside is 50 milligrams per milliliter, and outside is 50 milligrams per milliliter. So which side's higher? They're equal. So there's no high to low. Is there going to be movement? No movement. No net movement. There is movement, but there's no net movement. We'll talk about what that is. It's called dynamic equilibrium, and we'll talk about that in a minute. but there's no net movement. So they're equal. On this side, with this example, the outside was higher. On this example, they're equal. One uses numbers, the others just use symbols. You see how they can do this? Okay. So if I give you a problem, so now I give you a problem, you always have to ask yourself this question every time you answer these. And if I asked you, honestly, if I asked you in which, so this is, you were right, saying that yellow was higher and green inside and green was higher outside. You're all right on that. You're good. Now, what if I asked you in which direction is there movement for this one? I think I asked you and you guys answered you were wrong. In what direction will this move? The green. The green will move where? To inside. But you can't answer, the correct answer is, Mr. Mendoza, I can't answer that question. And then I can help you.
Ms. Perez wanted to know if Monet Ely will come to the office.
Ely,
She just went to the bathroom.
she just went to the restroom.
She's right there.
Oh, she's right. Well, somebody, why are you all telling me she's gone to the bathroom? Ely, does she need all her stuff? I don't know, there's always drama in there. Kids are just full of drama, I don't understand. Just want to come to school and go home. You know what I'm saying? I don't get. So anyways, inside and outside, these are equal. The equal, and which side's concentrate, which side isn't, okay?
So whatever is higher concentration, that's the one that
No, we can't tell. Why can't you tell? Why is the answer you can't tell? No, I did tell you this.
Can you repeat the question?
The question that you didn't ask, that I told you have to ask every time, that's right, does the solute,
Oh, cross the membrane?
that's right.
Oh. So,
In some way, somebody has to tell you if it crosses the membrane or not.
but
You have to answer that. The other question is which side's more concentrated? Hold on a second. No, I didn't tell you. I didn't tell you on purpose. I want to know if you guys are paying attention. So you can't tell in which direction the stuff's going to move because you don't even know if it's going to move. So if you assume what they'll do on a test, what they'll do on a standardized test is they'll tell you these materials can pass through the membrane easily. So they answer the question. They will answer the question. You have to read carefully, though. You have to read the question carefully. Yeah, yesterday I told you the answer was yes. I'm telling you now, they have to tell you, yes, the yellow crosses easily. The green does not cross. Now you got a problem if it doesn't cross.
So you told me that.
Yesterday, but every time you are asked the question, in which direction does the particle move, you have to ask, does it cross the membrane? Most of the time they will answer it in the question. But if you don't even know to pay attention, you're not going to know the answer. This is a very important question because it determines if you're doing diffusion or osmosis. Yeah.
So just to clarify, if it doesn't cross the membrane,
Then there's no movement of solute.
Okay, all right.
Then there's no movement of solute. Something's going to happen, but it's not going to be the solute. Yeah.
So, would that also happen when both of them are equal? Would it be no movement of solute?
Let me get to that in just a second. Good question. Go ahead.
Can the solute be like this? Like you said, the particle to be big, but then you use the particle to be
That's right, that's right. So it could be big, right? But if I tell you it crosses, then you know you don't have to worry about it. Either there's a protein in the membrane, or it's small and nonpolar, or something, right? Okay. Everybody good with that?
Wait,
Oh, okay.
when you say no movement, you mean no movement period or no, what's it called, no net movement?
No net movement. Very good. No net movement. We're going to talk about net movement in a minute, but I just want to make sure you understand no net movement. Yeah. Right. So far, we're not even talking about proteins. We're just saying somebody's gonna tell you somehow whether the stuff crosses or not. In every this is called simple diffusion. In simple diffusion questions, you're gonna be asked, you have to ask you have to ask, this is for you to ask yourself. And then you have to ask yourself this question. Usually the answer is already given to you in the problem. Are we all good?
One more question.
Yeah.
Okay, have we got to the part where it goes from like high to low? Because I had a question about that.
In a minute.
Okay.
Are we good?
Yeah.
Now let's assume that the answer here is yes. If you answered yes to this, then what do you have to figure out?
The direction. I mean,
Which is which we did, right? This is what we did here, right?
yeah.
So we did that here. We figured out which one's more concentrate for each of those. Now the next thing is we got, how do you, the last question is the actual question you were asked, which is what?
In
Which direction? So if the answer is yes, it moves across the membrane easily. And you know now which side's high and which side's low. Now you know which direction, right? Because in which direction is it going to move?
If we answered yes to the first
If you answered yes to the first question, so you know it crosses, it has to be able to cross, so it's not going to move. Then the next question is, which side's higher? We already answered that, right? There it is. There's the answer right here in this little area. Then the next question is what? In which direction? We know what in what direction? Well, no, what's high to low, high to low, high to low.
Well, I mean, Derek, I'm thinking like left or right or something.
No, I'm telling you high to low, because it could be left to right, it could be in to out, it could be all, the words are going to change, right? Just look at the problem, identify where it's high, where it's low, where it is high, where it is low, and draw the arrow in, draw it, so you're not confused, right? So I'm going to do it in color, but I would draw an arrow for the yellow one from inside to out. Why? Because it's higher inside. If I were going to draw it for the green dot, I would draw it from outside to in. Why? Because it's higher outside. High to low.
So they want to know both? I mean,
Yes, they could ask you a question where they would want to know both.
okay, so
Yeah.
what if they're,
So basically, the direction, after you figure out which side is higher and lower, you just say if we go in or out.
That's right. Yeah, now your answer would be the green particle would go into the cell and the yellow particle would leave the cell. That's what would happen. That's it. It's as simple as that. But you can't answer that. You can't make that simple answer unless you understood all three of the other things, right?
so what if there's just one concentration?
So what if the concentration is the same, right? What if it's 50 and 50? Well, there's no net movement. There's going to be movement. There's always movement. We'll talk about that in just a second. There's always movement. But the movements out is going to equal the movement in. The arrows will be the same.
It's called what?
We'll talk about it in just a minute. But are we clear to this point? When you look at equal concentrations, you're not going to be able to tell if anything's moving across the membrane because there's not going to be any change when you're looking at the beaker. You're looking at a beaker, you're not going to see any change. You're going to see this next week. Next week, we're going to look at an egg, which is going to be our membrane, right? And we're going to put it in a hypertonic, isotonic, and hypotonic solution. And we'll talk about what that is in a minute because that's where it gets confusing. If you don't understand this, you're not going to understand all those other topics, right? And then we'll put the eggs in those solutions, and you're going to see, in one solution, the egg will shrivel up. In the other solution, the egg will swell up. In a third solution, no change will happen. And you're going to measure the mass of each of the eggs. So you'll know if the mass has changed, then you know the stuff's gone into the egg. If the mass increased, you know the stuff went into the egg. If you see the decrease, the stuff left the egg. And if there's no change, then you know nothing. There's no net movement, right?
There's no change, that means it's equal.
They're equal, right?
No, Ms. Mendoza, what my question was, okay, there's one concentration. Some of it is inside and some of it is outside. If the
She means like if there's like one particle.
just one particle instead of two.
It's the same no matter what. 1, 2, 3, 50 particles. It doesn't, oh, you mean if there's just one single particle and there's nothing outside?
Yeah.
Then it'll be inside and then it'll be outside.
No, it's inside and it's outside.
But there's like no, it's like, say if you have green and yellow, but you only have green.
So what's I don't understand your problem. What's your,
There's an
You took out all
outside
those
and an
yellows.
inside, but it's only on one side.
Only
Yeah.
one particle, but for both inside and outside, like different concentrations.
Like what we were doing
Yeah.
yesterday.
one particle, 2 particles, 50 particles? It's the same thing. Figure out what the concentration is, which side's higher, inside or outside, for that particle. Right? And then decide which side's higher. Now you know, if it can move, it's going to move in that direction, from high to low. From its high, I keep saying it, from its high to its low.
Okay, so could there be, okay, so if there's one concentration, could it only be that something is moving from inside to outside? Or it has to be both?
Okay,
Like,
I don't understand. Look, if you have a cell, what are you saying? You have a cell, then what? You
okay,
have
so
one particle where?
inside and outside.
So there's many particles here, all the same particles, what you're saying.
Yes.
So let's say sugar, it's all sugar, all
Yes.
glucose. Okay, now what?
Okay, so now if there's less in the inside
So let's say 10 inside.
and 20 outside,
Can I use percent? 10% inside
yes,
and 20% outside. What's your question?
yes. Okay, so now it will only be the,
So now
The
we go back to the same process. I keep telling you, you have to learn the process. Learn the process. What's the process? 1, does the solute cross the membrane? I will say sugar, I'll say yes, it
Yeah.
does.
right,
Okay, so it crosses. 2nd, which side is higher? Which has more concentration?
the outside.
The outside. So this is higher. So what I do, every time I do it, I put high, up arrow for high, outside, down arrow for low, inside. Easy, easy peasy.
Okay, so my thing, I
So
got,
now wait, now what's the question?
which direction?
Okay, so now answer the question. Which direction does it go? From high, which is the 20, this arrow, to where? To low. You see, it's the same thing that we just did in the other problem. Yes?
Is anyone in here doing this?
We're in the middle of class. Can we do this during advisory, if that's okay? You're welcome.
So my question where I got confused, the second question,
Uh.
which is more concentrated, then I was thinking the one that's concentrated is the only one that's moving, correct?
Oh, I see. I think I'm beginning to understand what you're thinking.
Yes.
Let me get back to, write that question down so that your notebook can get an A. Because that was a good question. That's a question. Every time you write a question down, and I grade your notebook, especially if it's your question, it's obviously your question. You know your question is different from other people's questions. You're getting more points. That's called reflection. Hold on. You should be writing down your questions. How are you going to remember? Guys, you don't even remember what I say in class. And I record it and put it on the board. You guys don't, I keep trying to tell you, human beings don't remember well unless it's really important and you're writing it down. Even important stuff you forget. You forget where your house keys are. Heck, half of you forget your phones everywhere you go. I don't know why this is happening today. That's okay, go ahead.
Do you know where, Sinead is?
Let me pause this. It's my bad. All right, so I'm always trying, all right, so anyways, I'm glad you think my suffering's funny. All right, so in any case, the point, let's try to answer this young lady's question, and in order to answer it, we have to understand why this movement's happening. So, number 1, you learned in middle school, or a lot of you learned in middle school, that all particles are moving. Is that right?
Yeah.
You learned this idea of kinetic energy.
Yes.
And so particles are always moving. They're always vibrating. Even your table that you're writing on, the pencil you're holding, Even if you try to hold perfectly still, they're always moving. So if we took like a little circle and we magnified it, impossibly large, what we would see is that this particle sits next to this particle, and even if they were sitting still, they're vibrating. But they're not vibrating, they're moving in a direction usually. This was moving that way, that one's moving this way, random directions. Do you understand that? When you look at a glass of water and it looks perfectly still, those water molecules are spinning, they're moving to one side of the glass, they're moving to the other side, they're just bouncing around in that space all the time.
So when particles move, that's kinetic.
That is kinetic. When anything's moving, it's kinetic. Kinetic energy is energy of movement. Yeah.
Alright, so in middle school they told us that if it was a liquid, the particles move and move. It's a solid, move. So.
Well, I am so sorry that they taught you that, because that's not true. Shh, it's untrue. Now, maybe you misinterpreted, but that statement that you made is untrue. Let me say this. Kinetic energy is the energy of movement. All things are moving all the time. Now, what is true is that in liquids, they're moving faster than in solids. So when you're dealing, I'm going to quickly do this. I really hate going back, guys. You're stressing me out. When you're looking at a solid, the particles are really close together. So they're packed. So they don't have a lot of space to move. And there's all kinds of reasons for this. And we would call it, what we would do is call it, we would call it vibration energy, right? But there's not a lot of movement. But they're vibrating. They're moving, but there's not a lot of it. There's not a lot of space. There are these intermolecular forces, and we're not going to, we talked a little bit about that. Hydrogen bonding is an example of intermolecular forces. So forces that pull on each other between molecules. And so it keeps the stuff together. That's why water is liquid at room temperature, not a gas, because they're pulling on each other. So but they're also pushing, they're vibrating in every direction. And not just up and down, side to side, but also into and out of the board, right? Does that make sense to you? I'm not going to draw all these arrows. It's just ridiculous. But you get the point, right? These particles are moving all the time, even in a solid. The difference is in a liquid, they have more freedom of movement. Okay? So in a liquid, they fill up more of the space, right? If you have an ice cube, water we know is a little different, right? Because we know water, the solid takes up more space, but they're still locked together, right? So there's still that restriction of freedom of movement. The molecules can't move around, but they take up more space. So that's I don't want to be clear on that. Now these molecules, when they're solid, they can't move around too well, but that gives them a definite shape. That's how you get an ice cube, right? Can you get a water cube? Can you get a cube of water? No, because if the molecule can move, it's gonna move, right? It's gonna fill up the shape. If you put water in a triangle, what shape is the water gonna have?
A triangle.
You put water in a circle. What shape's the water gonna have? A circle. Where you put an ice cube in a triangle, what's the shape of the ice cube? It's a cube.
So that's how they do like popsicles and stuff?
Yeah, that's how they do popsicles. It fills up. You know how they have different shaped popsicles? Well, they just pour the liquid in, and because the liquid could take any shape, and then you freeze it. Now the molecules are frozen in that shape because they have that restricted movement. So I think that's where they were getting to in middle school probably, but no, everything's moving all the time. Quickly, question.
Okay, how does Jello move?
The what?
It's juggling.
God, do I really have to go over the states of matter? We don't have time.
No.
So Jello is called a colloidal solid, right? And so it is a solid. They do have restricted movement, but they have a little more movement than a true solid, right? So they have a little more movement, so that's where you get that kind of in between a liquid and a solid, right? We call it a colloidal solid. It's a suspension. Okay? Yeah.
So like you know how you said we drink water, you all drink water. When we drink the water, the particles in the water, does it like still have movement in our bodies?
Yes. Yeah. They just don't move, they move around. She says, and when you drink water, the particles in water, is there movement? Yeah, there's movement in everything all the time. It doesn't matter. That table you're writing on right now, it's vibrating like crazy. If it,
So what about
the,
the trash can?
everything, every atom in the universe is moving unless it's at absolute zero. And absolute zero is I don't even know if they have
So it's
anything.
moving when you're not touching it?
Huh?
So like the trash can moving.
The atoms are vibrating. They're doing this.
Oh, I thought you were saying only if you like.
It's not getting up and going anywhere.
I know
It's
that.
just it's just vibrating side to side, up and down.
If the atom is vibrating, why isn't the trash can?
Why is it what?
She said, then why is the trash can not vibrating?
It is vibrating. Very, very. You can't see it. You can't perceive it, but it is vibrating. Yeah, you're right. The whole thing's vibrating. Everything's vibrating.
So the walls.
Everything. Everything.
Everything.
Yeah. What is every? I mean.
Everything is everything.
So Santa Claus? No, because Santa Claus isn't real. But no, everything else, yes, it vibrates.
The moon vibrates.
I'm done talking to you. All right. So.
The moon.
All right. The kids just want. Yeah, they want it. And then you guys. Then we're all stressed out. Everybody's upset. So you know what I'm going to do? I'm going to finish this, and for homework, I'm going to post this thing. It's going to be really long. You obviously don't have to watch the whole thing.
Right.
But the part that we weren't able to get to because, I'm not going to say it. So you need to make sure you get, because we're on Monday we're starting off we got to move faster now.
Right.
So now here, kinetic energy is vibrating, the particles are vibrating. Now let me be very clear. If you have this opening, and that's the only way things are going to get from one side of the membrane to the other, there's some kind of opening. And you have this opening, and you have these particles over here, they're really concentrated. They're always bumping into each other. They're moving in every direction, right? Because it's in a liquid, it's not a solid. If it's a solid, they're not going to flow, right? They're not going to diffuse. Solids don't go anywhere. We're talking about liquids and gases, right? So these atoms or molecules are in a solid. They're part of a solution, and over here there's less of them. Now there's water all around them. They're moving all the time in every direction. So some of these particles are going to come over here, right? You're going to get one randomly moving over there. This is all random. Then you're going to get some of these. We're going to bump into each other and move in this direction, right? But there's more stuff over here, isn't there?
Yeah.
So what are you going to get? More stuff. That's right. So if we do the right movement, the right direction, the direction to the right in green, and the direction to the left in yellow, in which direction is there going to be a net movement? Where's most of the movement going to happen? To the left. So if you're looking at your egg, or you're looking at a die, let's say we drop a die in there. What are we going to see? That the stuff is moving to the left.
Is it like that because it's more on the right?
That it's like that because there's more on the right than the left. But that's why things move from high to low, because you have so much stuff over here at the higher concentration. You have so much stuff at higher concentration, they bounce around. They're all bouncing at the same. By the way, what determines how fast they move? This thing.
Temperature.
So how fast things move in a liquid or a gas all depends on temperature. Are we clear? Because temperature is a measurement of get this, write it down. Temperature is a measurement, it's a number that means the average kinetic energy. Temperature means the average kinetic energy. The average amount of movement. So now if I draw something like this, and I draw stuff like this, and we have that openings in the center, we know that is there movement in this direction? Yes, there is. Only a little bit. But is there a movement in that direction? Sure, there's a lot. Now, what happens if both sides are equal? No, there is movement. There's movement in this direction. But then what happens? In the other direction, you have what? Equal movement. So when you're looking at it with your eye, because our eyes can't see atoms and molecules, right? When we look at it with our eyes, what do we see? We see no movement. We don't see the movement, but we know that they're moving back and forth. It's just for every one that goes to the right, then one goes to the left.
Oh, that's what keeps it balanced.
That's good. Good. Let me write down what this is. This down here is called dynamic. Dynamic means moving. They're moving. Static means not moving. Dynamic equilibrium. Dynamic equilibrium means there's movement in one way. For every one that goes to the right, one goes to the left. We call it dynamic equilibrium. Equilibrium because they're equal movement. Dynamic, it means movement, right? So equal movement, dynamic equilibrium. So there's no movement when you're having dynamic. Now, if you have over here, this one, there's more on the left than there is on the right. So because there is movement in both directions, you see that, right? But the overall movement is to the left, is to the right here. That's the movement we notice. That's the movement we can measure. We can't measure movement in dynamic equilibrium because for every one that goes one way, the other one goes the other, and we've done this. We could do it with, if we put the same size particles on the right. Let's do this thought experiment. What if I put a radioactive element, radioactive potassium on this side, and that gives off a certain frequency, let's say. And over here I put radioactive sodium, all right? And that gives off another frequency. So they're basically the same charge, right? And so if I measure on the right, I wait a little bit, what I'm going to find is I'm going to have equal amounts of potassium on this side that I do on this side. Equal amounts after a while, because what's going to happen is for every one potassium goes to the right, there's one that's going to the left. And one sodium goes to the right, there's one goes to the left. So after a while, we will get to dynamic equilibrium. Yeah.
Do you think it's possible to tell me, like give me an example of this, like in a real life situation?
Well, we're gonna dynamic equilibrium. That's one of the labs we're gonna do next week. You're gonna get water in a jug, and you're gonna get a cup, and you're gonna actually do this. You're gonna move for every one cup that goes one way. You're gonna put one cup's coming in, and we're gonna see how if the volume ever, if the amount of water ever changes. And the answer is no, it won't change.
Why did you say this?
Because you'll see it. It's not that simple. It's a little more complicated. But you'll see it. You'll see it. You'll see dynamic next week. Hopefully, God, cross your fingers. You'll see dynamic equilibrium. You'll see the egg. We'll do the egg because the egg is going to take time, and we're just going to measure it once to a chicken egg. And we're going to do yeah.
We're doing a chicken egg?
Yes.
Yes, we're doing a chicken egg. So we have to use our gloves because we don't want anybody getting sick. All right. Well, I think we did a pretty good job today. I love your questions. I just hope you guys. I hope you guys watch the rest of this video because it's going to be hard to do everything we have to do next week if you don't.